proteins that are embedded within the membrane have to have charge distributions that match their location as partly within a membrane and partly exposed to the water and ions and proteins and other molecules in the intracellular and extracellular side of the membrane. I'll start talking about these proteins by starting with membranes, which I normally draw as two lines or just a single line, but now I'm going to put in the phospholipid bilayer with the charged heads and the hydrophobic tails on the inside. These are carbons, long chains of carbons with hydrogens, and these are neutral. So if we had a typical protein spanning a membrane with the amino acids here, and the amino acids would be about this size on this scale. This particular protein has 7 transmembrane regions, common to or a large subset of protein. So 7 transmembrane domains, and each one is a string of amino acids with the neutral interior of the membrane, this region, or with the extracellular and intracellular sides of a membrane. So on the inside, we need amino acids that are neutral, interacting with the neutral lipids. In this case, I've drawn them on both sides, but of course our protein is three-dimensional, and so the plasma membrane is surrounding it on all sides. On the other hand, on the outside and the inside, there are water molecules that have an uneven distribution of charge. They are polar. They're not net negative or positive, but the oxygen tends to hold the electron a little bit more strongly. And water molecules here, and there are also ions. And the parts of the protein that project out above and below the membrane surface need to have charges in order to interact well with those. So if I put an amino acid here, some of these would need to have a positive charge or a negative charge in order to interact well with the water and other molecules on the inside and outside that are charged or are polar. Now, not all of them are going to be charged. In fact, about 30% of the amino acids that are on these projecting regions would be charged. So if I highlighted the different amino acids that are on the outside, some of them would be charged and positive, and others would be charged and negative. In fact, the outside and the cytoplasmic side, the inside, tend to have different distributions of positive and negative charges, and you can predict which face a particular protein is by knowing the amount, although we won't do that today. So the key points are that you would expect this inner region of the protein to have mostly neutral amino acids, and in fact, on this inner region of a typical protein, only about 3% of the amino acids are charged. And those that are charged tend to help hold the protein's internal shape. So you might have a negative charge here and a positive charge here. They're neutralizing each other, and they're tending to hold the protein together at that spot. In contrast, the part of the proteins that are in contact with the cytoplasm on the inside or the extracellular matrix and fluid on the outside, they tend to have charged amino acid, and as I mentioned, about 30% are charged.